The experiments described in this video, recording action potentials with the neuron spiker box, use invertebrate animals, which can be easily acquired from a pet store, bait shop, or ordered online. All of the animals are anesthetized prior to making these recordings. Firstly, it's just cool to be able to see and hear the electrical signals that neurons use to transmit information. After talking about the molecular basis of the action potential and how an electrode can measure these changes in voltage, now your students will literally be able to see these action potentials in neurons. This ability to eavesdrop on the messages that neurons are sending has been critical for forming our understanding of how the brain works and which parts of the brain process what information. Recordings from cockroach legs are generally the simplest to start recording with your spiker box, and these cockroaches' legs will regrow given about half a year. This is also a chance to see one of the basic concepts of how the brain represents information in the form of spikes in neurons firsthand. And this is the concept of rate coding. So in this case, the sensory responses that you're recording from the cockroach leg will reflect the strength of the tactile stimulus that you provide to the leg. A stronger tactile stimulus leads to more action potentials being fired, and that is the fundamental concept of rate coding in neurons. In addition to recording from these sensory neurons, you can also deliver electrical stimulation to the nerve, activating motor neurons and contracting the muscles, causing the leg to twitch. In fact, you don't even need the spiker box to do this stimulation experiment. This ability to directly electrically activate neurons has been used since the late 1800s, so almost 150 years, and has contributed a great deal to our understanding of the localization of functions within the brain tissue. This can also be a window into discussing a very early debate in neuroscience, which is the question of whether a neuron's signals were electrical or chemical. Electrical stimulation, of course, provides evidence in favor of using electrical signals. And as we now know, the answer is actually both. Electrical as the action potential moves within a neuron, and then chemical communication at the synapses between neurons. With the Spiker Box Pro, which will allow you to record two signals at once, you can do experiments to measure the conduction velocity of an action potential. That is, how fast the action potential moves along the length of an axon. Earthworms are great for this. They have quite a simple set of sensory nerves that are well suited to recording the same action potential from two different electrodes within a single axon. The Spiker Box Pro and accompanying software will allow you to very precisely measure the time delay between when an action potential passes one electrode and the next, which combined with a measurement of the distance between those electrodes is going to allow you to calculate the conduction velocity. This ability to measure conduction velocity then opens up multiple experimental options. For example, you can compare the velocity of the action potential in different sensory neurons, or as a function of changes in temperature. More detailed guides on performing each of the experiments that I just mentioned are available from Backyard Brains, and I'll provide links to all of those in the Neurophysiology Resources document.